Good morning, everyone. Yes, I am Elisa Lovecchio. I am a visiting postdoc at the National Oceanography Center of Southampton in the group of Stephanie Hansen. The work that I will present today is mostly based on a modeling study that I developed during my PhD at ETH Zurich together with Niki Gruber, Matt Munich, and Zuhair Lahar, and which was further extended in the last 2 years thanks to a fruitful collaboration with Giulia Bonino at CMCC in Italy. At the end of the main part of the talk, I will also shortly talk about my current work together with Steph Hansen, Filippa Carvalho, and Nathan Briggs at the National Oceanography Center. So the title of my talk is 3D cycling of organic matter in upwelling regions, driven by small scales and impacting on large scales. And since I will be talking mostly about the organic carbon, let me start with the marine organic carbon cycle. As most of you know, the organic carbon is produced in the euphotic layer of the ocean by phytoplankton. The euphotic layer is roughly the first 100 meters of depth in the ocean. It's produced through the uptake of inorganic carbon and nutrients by phytoplankton. Great part of this primary production gets exported vertically below the euphotic layer, where it gets in great part respired, remineralized into the inorganic constituents. And this vertical displacement of the organic matter and of the constituents is the reason why this is also known as biological pump, as it pumps the organic and inorganic material at depth. As we can see from this picture of chlorophyll from satellite data, not all the ocean is productive in the same way. There are some parts, like the coastal oceans, which are extremely more productive than others. And among these, the eastern boundary upwelling systems are a great example of very productive coastal regions. And this is because of the resurfacing of nutrients along the continental coast. Let me add this distinction between the low productive open ocean and highly productive coastal ocean in the biological pump scheme. Generally, or very often, the biological pump is described as a one-dimensional vertical process that is dominated only by the vertical relocation of the organic matter through vertical export below the euphotic layer. In this scheme, each column of water must remineralize at depth as much organic carbon as it produces in the near surface. So each column of water is sort of isolated from the others. And it must balance, the net community production must, when you integrate it vertically, must add up to 0. However, there is plenty of observations in which we see that sinks exceed sources in the open waters, especially in the low productive regions. So we must look for extra sources of organic carbon. I'm going directly to the spoilers in this talk because we're talking about biogeochemical modeling. And I'm showing you here the vertically integrated organic carbon concentration in the Canary upwelling system from a 3D fully coupled physical biogeochemical model. And I'm doing this because I want to show you how also just looking at the dynamics of what a model can show you can give you a first understanding of what's going on and can give you an answer to questions that that you want to address. So just look at the organic carbon concentration in this biogeochemical model, and you can see that at the upwelling coast, the organic carbon that gets produced at the upwelling coast gets exported laterally. There is a strong lateral transport from the productive regions towards the North Atlantic gyre, so towards the low productive regions. And this happens not only through mean circulation processes like coastal currents, Ekman transport, but also thanks to very fine small-scale structures such as upwelling filaments and mesoscale eddies offshore. So going back to the biological pump scheme, I added here the possibility of lateral transport of coastal carbon, which does allow the coastal ocean to be a net source for the organic carbon and the open ocean to be a net sink for the organic carbon. And the focus of my talk will be exactly this. So I will talk about the lateral transport of organic carbon and focus in particular on the Canary upwelling system. Towards the end of my talk, I will talk a bit about the Benguela upwelling system. That is what I'm working on at the National Oceanography Center more recently. The model of the Canary upwelling system that I used during the PhD is a fully coupled physical biogeochemical model. I used ROMs for the physics and an NPZD model for the biogeochemistry. The NPZD model is a nitrogen-based model. It's a relatively simple model because it has only one type of phytoplankton, one type of zooplankton, and it has two classes of detritus, one fast sinking, the large one, and one slow sinking, the small detritus. These four pools constitute together the organic carbon pool in the model. We ran this coupled model onto the Atlantic telescopic grid. This is a full it's a grid that covers the entire Atlantic basin, and it has a strong grid refinement towards the North African coast. That is the focus of our study. So it has an eddy resolving resolution in the Canary upwelling system. The model was run in climatological mode because we want to look at the mean state of the system. And what we used for the analysis are 24 years of analysis output in the form of B daily means. So the first question that I want to address is what is the overall magnitude and what is the impact of this offshore flux of organic carbon? What I'm showing you here is a plot of the zonal flux of organic carbon from the model output. This zonal flux was calculated as the product of velocity and concentration at model runtime. The plot that I'm showing shows the 100 meters integrated. So the zonal flux is integrated in this plot throughout the euphotic layer, so throughout the productive layer of the ocean. As you can see in this plot, the widespread blue signature indicates that the organic carbon is transported mostly east to west, which means from the North African coast to the open waters. And in fact, if we go to average over the entire upwelling system, this flux, we see that on average, it is indeed a negative signature over the entire Canary upwelling. So I'm drawing here the 100-kilometer offshore line that you can see rounding in front of the African coast. This is the boundary of the region that is most directly impacted by the upwelling. If I integrate the lateral transport through this line, I find that as much as 1/3 of the entire net community production in the first 100 kilometers crosses this line, and it's exported towards the open waters and can eventually be remineralized there. In fact, if we integrate vertically the net community production in the entire system, think back about that biological pump scheme that I showed you before, you can see that the area next to the coast is a net source of organic carbon. It has when we integrate it vertically from the top to the bottom of the water column, it has a positive signature in net community production. While the offshore regions remineralize more organic carbon than what they locally produce. And this is true for a very, very widespread area that reaches basically into the North Atlantic gyre. Now we want to know how big is this extra organic carbon availability that reaches these regions. And I divided the entire system into 4 offshore boxes. Each one of these boxes, you can imagine it as a 3D box that is as deep as the euphotic layer, and it extends offshore up to the next line that you see here. These lines, each box is more or less 500 kilometers of offshore in the offshore direction. So the first one ends at 500 kilometers offshore, then 1,000, 1,500, up to 2,000 kilometers offshore. For each one of these boxes in the euphotic layer, I calculated the net community production and the export at the boundary of the euphotic layer. So how much carbon gets transported from the euphotic layer to below. If I make the difference between exported carbon and locally produced carbon, I actually get positive results, meaning that each one of these regions is exporting more carbon than what it locally produces. And it's up to 40% more than what it's locally produced between 100 and 500 kilometers offshore, which is also the most heterotrophic area overall. Now we want to know if this can be explained by the local transport of organic carbon. And so an easy way to do this is to literally calculate the difference between the amount of organic carbon that enters and exits each one of these boxes. And doing this, we can see that this amount of organic carbon that gets basically released inside the box by the offshore transport can explain a great amount of this extra export from the euphotic layer to below, adding up to 30% of the local production in the most close box to the coast, and then going down. And it can explain up to 60 to 80% of the extra export down the euphotic layer, up to 1,500 kilometers away from the North African coast in the Canary upwelling system overall. There are sub-regional differences in this. And in fact, you can see that the central part of the Canary upwelling system, for example, has an even more negative signature in the vertically integrated NCP. I don't have time to go into this right now, but if you're interested, you can look into the publication on Biogeosciences. The second part of what I want to discuss regards the role of mesoscale eddies and filaments in the transport. And these really can show you the power that these biogeochemical models have in terms of how much information you can get from these type of models. We want to divide the total transport into how much of this transport is carried on by cyclonic eddies, anticyclonic eddies, and upwelling filaments. In order to do so, we identified the eddies using a sea surface height-based algorithm. This was developed by Fagmos et al. In 2015. And we also identified the edge of the upwelling filaments that are these very cold water signature structures that develop from the coast and propagate offshore. In order to identify the upwelling filaments, we actually used a sea surface temperature algorithm that was developed during this study. So once we know what is the outer boundary of each one of these structures, and so what is the area within each structure, we can calculate the flux carried on separately by each one of these types of structures by multiplying the organic carbon concentration and the velocities in each one of these. So the total flux is basically the sum of the cyclonic eddy flux, anticyclonic eddy flux, filament flux, and all the rest, the non-mesoscale flux. And this is how I'm going to decompose the total flux in the next slide. So imagine this is the total offshore flux. There are some components that can go actually onshore, and so they will have a negative contribution to the total flux. And I also decomposed what I showed you before, so the divergent, how much organic carbon is released in each one of those offshore domains that we discussed before by each type of structure. And what we can really see clearly from these plots is that filaments have a very important role in the offshore flux in the near shore 500 kilometers up to 1,000 kilometers. At 100 kilometers offshore, so just at the boundary of the upwelling area, filaments drive 80% of the total offshore flux. So a really, really big part of the total flux of organic carbon. And they also have a very important influence in terms of how much organic carbon they add to each one of the offshore domains that we looked at before. So up to 1,000 kilometers offshore, they are still the major component that they are responsible for most of the added organic carbon up to 1,000 kilometers from the North African coast. This means that very small structures such as filaments, which are extremely narrow, have a very important role in the lateral relocation of the organic carbon. If we look instead at the eddies, anticyclonic and cyclonic eddies, we see that their contribution is smaller. However, eddies are slow structures, especially if compared to filaments. And this is the reason why their contribution to the flux itself is smaller. Despite that, they're still very organic carbon rich, and they contain more organic carbon than how much area they actually cover in the model. So they are enriched in organic carbon compared to the surroundings. And this is especially the case for cyclonic eddies. So eddies are slow. However, as you can see from these plots, they're far-reaching, and they are important carbon reservoirs for the offshore waters. As a last point, I want to discuss the temporal variability of the offshore flux of organic carbon and its impact on the open waters. And this is the very last, the most recent part of the work that we did with this model. And I'm going back to the initial animation that I showed you before. But this time I would really want you to focus on how the organic carbon concentration next to the North African coast changes in time. So the Canary upwelling system is actually known to be the one that has the most intense seasonal variability among the major upwelling systems. So as you can see here, the organic carbon concentration varies in time, and it peaks in some seasons at different latitudes. And I want to focus right now mostly on the central part of the Canary upwelling system, because it's the one that shows the highest organic carbon concentration and also the farthest reaching transport of organic carbon that really gets far into the open waters and reaches the farthest area of our analysis domain. What I show you here is a Hofmöller diagram of 5 years of simulation on the 2-day mean output of the same variable. So it's still the organic carbon concentration. You can really see that there is a spring peak in organic carbon concentration near the coast that gets transported offshore towards the open waters. In every season, filaments continue to be the driving force of the organic carbon relocation. And in the first 500 kilometers of shore that are covered by the filament transport, so in which filaments really have an important role, this seasonal peak in the organic carbon concentration reaches this range of offshore distances nearly immediately, so within the same season. But if we go more offshore towards the open waters, it will take more time, and it's both because of the farthest distance, but also because the processes that drive this transport offshore are slower, such as eddies and such as non-mesoscale transport. This means that the seasonal peak in the organic carbon production near the coast will hit at some point the open waters, but it will take some time, and it will arrive there with a certain delay that could get as big as 1 year time. As a result, when we look at the seasonality of the net community production integrated throughout the entire water, so we look at what areas of the sector of the Canary upwelling system are in the net sense are producing or using more organic carbon than what they locally produce. The result of this plot will be a combination of local processes, such as, for example, the North Atlantic spring bloom or the stratification of the water column, and of the organic carbon that is getting there through this lateral relocation of the organic carbon from the North African coast. And so they will be hit also by the seasonality of the processes that happen actually thousands of kilometers away. In other words, the organic carbon flux transfers offshore the seasonal variability of the nearshore processes, such as production, Ekman transport, upwelling, and modulates in time the biological activity of the open waters for thousands of kilometers. So as a summary of this part of the talk, we saw that our model could show that 1/3 of the coastal production gets exported towards the open waters, and it has this transport has a very long range impact. So the biological pump is clearly a 3D process in which coastal ocean and open ocean are coupled. We also saw that most flux, especially in the nearshore, is driven by narrow structures such as coastal filaments. And this is important, for example, for coarser models that cannot resolve these scales. And they may need to include a parameterization of the filament flux to actually capture the dynamics of the organic carbon cycle in the ocean. We also saw that eddies, even though they're slow, they are far-reaching, and they are rich in organic carbon in the open waters. So also in this case, also observation studies must take into account a certain level of variability in the organic carbon flux. And finally, we saw that the coupling in time between the coast and the open ocean means that the organic carbon relocation can actually transfer offshore the type of variability that affects the coastal waters. In the case of the Canary upwelling system, we're talking mostly about seasonal variability, but it could be a different type of variability for other upwelling systems. And thanks to this organic carbon relocation, this type of variability hits the open waters at very large distances from the coast. So I am now going to give you an outlook of this work. So I want to show you how we can use these high resolution models, coupled models, for different purposes. In this case, this is the work that I'm developing right now at the National Oceanography Center in the group of Stephanie Hansen. So Stephanie, together with Filippa Carvalho and Nathan Briggs, collected some data in the region of the Benguela upwelling system. This region is actually very interesting because it's really an example that puts together all that we spoke about till now. There is Ekman transport because we are in an upwelling area, there are eddies, there are filaments, there is a frontal region, and the area where the gliders were deployed that is indicated by this small dot is actually hit by all these types of dynamics. As a result, the glider data show these very intricate dynamics, and it's difficult to understand what is going on just looking at the glider data. And this is really where models can come in to help. So I'm showing you here the oxygen concentration from the glider data. And you can see that there are these shallow, low oxygen anomalies that show between, let's say, from 50 meters depth and below up to about 300 meters depth. And they show us these grayish, almost white blobs of water that hit the glider. And they seem to appear and disappear. And in the meanwhile, we also see oxygenated water, actually, that is located at the glider, that reaches the glider position. So we really want to understand what's going on here. And to do so, I developed this model setup that is a higher resolution model than what I showed you before. And it's also smaller. So it's only this square around, really centered around the glider position. It's still a fully coupled physical and biogeochemical model. So what I will show you here is an animation of the oxygen concentration sliced at 100 meters depth in the domain that I showed you before. And I really use the same color scale so you can compare what the glider saw and what the model sees in terms of oxygen concentration. The model is a hindcast model, so it's the same year. I used 2018 forcing and 2018 boundary conditions. However, due to the chaotic trajectories that characterize the ocean flow, it's impossible to reproduce exactly the same type of variability. What you will see is something that will reproduce statistically a type of variability that is potentially as similar as possible to what the glider actually saw. And here you can see that the low oxygen concentration water that comes from the northern part, this is the Angolan part of this system, hits the glider. And sometimes instead other structures with higher oxygen concentration arrive from the southern area. So now we can use this model. First off, we can understand why we see these alternate patterns in the oxygen concentration. And we can also deploy, for example, floats in the model and track what kind of waters hit the region in which the glider was deployed to collect the samples. And as a really closing, the really closing part of my talk, I just want to let you know about this project and invite you, if you're interested in Eastern Boundary Upwelling System Science, to join these webinars. They're called EBOS webinars. It's an international project that started 2 years ago. It was initiated by early career researchers, and we are now 8 people, 8 researchers keeping this project running. And it was started with the idea of making scientific knowledge exchange about Eastern Boundary Upwelling System research more inclusive and more sustainable. Here it's just a sketch of the affiliation of the speakers that presented their works in these webinars. We had so far 30 webinars. It's a totally free project. We have, it's really only science oriented. There are 15 webinars recorded on the YouTube channel of the project. So if you're interested also in seeing what kind of presentations already happened, And I want to talk about this also to send a positive message, because in the middle of the pandemic, we find now ourselves having to communicate only virtually. But this is a project that started basically 2 years, a year and a half before the pandemic hit. And the positive message is virtual communication of scientific knowledge can really work. And it can actually broaden your network of collaboration and reach far where you would have not probably gone in person. So if you're interested in this type of research, follow these webinars, or I could also say I encourage you to start your own webinars on the topic that's really most of interest for you in your research field. And yes, so with this, I really thank again, Francisco, for inviting me. I thank my current research group at NOC, and special thanks to my co-authors during the PhD and also in the recent years. So thank you very much. And if you have any questions, I'm very happy to take it.